really like to be among you, but travel restrictions do not allow. Hopefully, I can see you all in person next year. Just wanted to say I feel privileged to be among you all. It is representing the industry that has a lot of bright colleagues is a huge undertaking. So I hope that what I'm going to talk about and speak to you all provides some insights and some value. I attended. I am based in Montreal, so I got up early and attended all the talks, and I believe I am reassured because what I'm going to say is simply a continuation of what you all have said very, very elegantly just a few minutes before. So Farzan Ali, I am heading the Patient Health Impact Group at Pfizer for hematology, colorectal cancer, and melanoma. Patient Health Impact is a health economics, outcomes research, and market access all together. So those are the teams that I lead. Maybe we can go to the next slide, which I believe is the usual disclaimer slide. The views are presented, though they may be aligned with Pfizer's, they do not represent the views of Pfizer. Those are my views as the presenter. We can go to the next slide. And basically what I wanted to talk to you about today was the use of real-world data. I think we have talked about real-world data a lot. What I wanted to do today was give a few other angles. Some of it might be some repetition, which is good, but maybe give some angles from an industry perspective on why we think real-world data could solve a few problems or challenges that we see. So first of all, I just want to say I am a big fan of randomized controlled trials. I love randomized controlled trials. I think randomized controlled trials, they provide robust and reliable information about the benefit of medicines. They also, of course, there could be badly designed RCTs, but usually RCTs minimize the sources of bias. And let's face it, RCTs are the gold standard. So when you get results, when you get an output from an RCT, you do understand what it means. Everybody understands the analysis plan, the statistical plan, and what the results are saying. So if we go to the next slide. However, like I said, I will reiterate, RCTs are amazing, but I think there are some issues. Ideally, we would do RCTs all the time, but as we realize, and I think some of you know, RCTs sometimes have practical and ethical challenges currently, especially in cancer where treatments are evolving, the landscape is very dynamic. Provide one tangible example of, let's say, ethical concerns. For those of you who remember when ALK inhibition or ALK inhibitors came on the market, in lung cancer, when you saw a very, very strong response in a phase 1 with an ALK inhibitor in patients who had ALK mutated non-small cell lung cancer, then doing a second trial, a pivotal registrational trial for ALK positive patients, for ALK positive lung patients, did hold some ethical concerns because you identified ALK positive patients. You knew that in phase 1 they had amazing response, but you're giving them something that is not targeted. It's just general chemo. So I think as we talked about, I gave the example of a lung cancer. I think it's the same thing in hematologic malignancies where we are discovering a lot of gene mutations and biomarkers, and you see amazing, significant results in the phase 1, and then you go on and you try to do a randomized control trial, then these ethical questions abound. I think the other issue is that as we all know, maybe 100 years ago, it was called blood cancer, right? Would say blood cancer. Now we know there's many types of blood cancers. There's not just one general blood cancer. And actually, there are many types of AML. There's not just one AML. And maybe in a few years, there'll be many type of subtypes and subtypes of the different subclasses of AML that we know now as we understand the biomarkers and the genetics and so forth. So there are a lot of different types of cancers out there. And as we slice and dice and we understand patient populations better and how to treat them, we come with very, very niche populations. Already hematologic malignancies are rare. Imagine if you look at subtypes and so forth, they become even more rare. So the practical challenge of even recruiting patients in your active arm, that is difficult. Imagine having another one for your control arm. So those are a couple of things where a couple of aspects or challenges that you could identify with RCTs. If you go to the next slide. And that's why we see a rise in what we call SATs. So what is a SAT? A single arm trial. We have at Pfizer used single arm trials. I know that our other manufacturers are using single arm trials. This presents, the graph on the right presents HTA submissions. So health technology assessment submissions, not necessarily the regulatory side, but you can see there's a rise in single arm trials because of some of the challenges with RCTs. However, if you see on the left, and by the way, one important thing is that 65% of SATs currently submitted to HTAs are in oncology and or hemato-oncology. So that's quite interesting. Unfortunately, if you look at the use, well, so when you have a single arm trial, what you're asking is, okay, this is great. I do see the challenges of an RCT, but this single arm trial, how good is it? The results versus what's out there. And sometimes what you can do is use real world data, right? And that's one of the things I wanted to discuss here. But what we see on the left-hand side, the one thing is that only 20% of the single arm trials are complemented with real world data. And if you go to the next slide, and we can get to some of the challenges. Oh, I'm off. We can get to some of the challenges of why that's the case. I think I was really glad that AML was brought up a few minutes ago. I think another part, so we talked about RCTs being a challenge, the use of single arm trials. Let me propose another angle to this, which is making RCTs quite difficult, and where real world data could come quite in hand or quite beneficial. When you have a very dynamic and evolving treatment landscape, like in AML for many years, I would say the innovation productivity was stagnant. But in the last few years, this is amazing news for patients. There's a lot of innovations that have come on the market for AML. So if we go to the next slide. If you have a very dynamic, rapid treatment evolution and you're doing an RCT, I think, just a cartoon here to represent, we showed AML. It could be probably any hematologic malignancy right now because there's a lot of research being done. If you start your RCT, let's say you forget about the practical and ethical challenges there, you have solved them. You do start an RCT and at the time that you're starting the RCT, the standard of care was treatment A. So you have your new therapy, you want to compare it to treatment A. And this is from personal experience. This has happened to one of the assets that I have been working on. While you're doing the trial, because the trial takes some time to read out, there's treatment B that comes, treatment C that comes, treatment D that comes. And the question after when you have read your trial as read out later on is, okay, it's great that you're better than the standard of care that was there a few years ago, but how good are you versus treatment C? And I think if we just click maybe another time, I think one way where real world data can come in handy is that it can, if done correctly, because real world data is not perfect either, if done correctly and if we capture it correctly, we can actually capture treatment A, treatment B, C, D, any other thing that comes in between. So that is one of the benefits potentially, idealistically. So I'm trying to be utopic and ideal here, but that could be one advantage of real-world data to capture the rapid treatment evolution. Another example, if you go to the next slide. Another example is, and I think Dr. Pignati mentioned this, so I was super happy. This sounds like I made these slides after everybody talked, but actually these slides were made a few weeks ago. So we have a treatment, and this also happens often. You want to do an RCT, and but the comparators are different. And this is also from personal experience. In the US, it could be treatment A, which could be the same in Germany. In France, could be B. In Japan, could be E. And because I am from Canada, I always pick up on Canada. So I'll pick up on Canada because we'll say they don't have the new treatments. It's only palliative care. So how do you go about this? How did you cannot have a humongous clinical trial that's going to read out in 20 years. That's not very patient-centric for patients who are waiting. So if we click another time, I think if, again, done correctly, you can have different, if you have a global real-world data control arm, you can capture all these comparators and compare your new therapy to them. So if we go next. So we talked about the challenges of RCT, and I think, we would sit here and, a lot of you much more brilliant than I am could talk about a lot of the challenges about real-world data. Let me propose a few because real-world data is not perfect, at least as it currently stands, which could explain why it's not used a lot to complement or supplement or support the single-arm trials. First of all, there's missingness of data. I think I'm also super excited that we talked about patient-reported outcomes. At the end, what we are doing here is for patients, and what counts is that how patients are doing. What is their functionality? What is their quality of life? And Guillermo also mentioned this. A lot of us know it. Salen also brought it up, and Tamash will probably bring this up as well. He's just after me, is that PROs are missing in the real world because we don't capture them in the real world. So that is something that usually we don't capture them. I'll be super excited to find registries that do. I'm giving MRD an example. There's many other examples of clinical endpoints that are missing. Protocol-driven approach versus real world. So for example, in a clinical trial, you might see the physician or the nurse or the research center every month or even more, every week, maybe every 2 weeks. You might see in the real world, depending on the tumor and the hematologic malignancy, you might see the physician every 3 months, every 6 months. So you get a more precise assessment of PFS in a clinical trial, while you get a more crude, rounded up PFS estimate in the real world. Matching methodology, I'm not a stats expert, but I'm naive. Comparison is no good. You have to match and wait to actually make sure that the patients that you have in your active arm or in the real world arm represents the type of patients you have in your active arm. And magnitude of benefit is important. If you have 2 months of PFS, additional PFS in a clinical trial setting, it's not great, but everybody would accept that is probably true. You're getting a benefit. If you have 2 months of benefit versus real-world data arm, there's a lot of questions because that could just be noise. So the magnitude of benefit is important. Now, I do see, without naming any names or any countries, that there are some countries that have put out some suggestions or recommendations on what that magnitude of benefit should be, and sometimes it's in the realm of 10 times. And I think sometimes that 10 times becomes a bit unrealistic based on the hematologic malignancies and even life expectancies of some of these patients based on age and so forth. And ability to pool, I think we are at an Harmony AGM. It's such an exciting consortium and platform. I mean, this is what Harmony does amazingly, and that's why we are so proud of Pfizer to be part of this. They harmonize data. This is an opportunity for us to all come together and pool data, because when you are talking about rare hematologic malignancies, one registry may not be enough. Next slide. So this is my conclusion, and I sincerely hope that I brought a few angles and that I brought some value to this. First of all, RCTs are a challenge. I do like RCTs. I just want to make sure that people don't get the idea that I don't like RCTs. I love RCTs, but there are challenges to doing RCTs. I think if anything, COVID-19 has shown us, either with the vaccine or the oral pill, that we have to adapt ourselves. And right now we are at an amazing opportunity for patients where we are coming with a lot of innovations for hematologic malignancies. I think we also need to adapt and think about how we're doing trials and how we're bringing evidence to keep up with the speed of science. The use of real-world data, pre-approval and post-approval are great. I think I will not belabor this point. Even post-approval, I think there's a lot of value. We have to, my call to action, if I can permit myself, is that let's all work collectively to make this happen. We know RCTs have got challenges. We know that real-world data currently is not perfect. How do we make it so that we have an opportunity to leverage real-world data to accelerate R&D, and we can have clear guidelines from a regulatory HTA that captures things that are important for patients, and that pharmaceutical companies know what the guidelines are, so that we can do it correctly. And I would say the last point is that it would be amazing if every patient that we have in front of us that we treat not only is amazing news because that patient is being treated, but if that patient becomes part of a thing that much bigger, they become data for future innovations to come. And if we can consider every patient that we are treating, the data that we enter in an EHR and so forth to be as complete and as robust, then imagine what that can do. The pool of data, the ongoing data that we get, we might see a path to chronicizing or curing some of the cancers at hand with the speed and the massive pooling of clinical and real world data. We might see the optimal treatment paths for the different patients that we want to treat. So thank you very much. I hope I kept on time, and hopefully I will be there in person next year.